This is Biology Unit 6 revision video looking at useful microbes such as microbes in making yoghurt and brewing. So we've looked previously at how pathogens and microbes can cause disease, but as well as being dangerous to humans, they can also have a lot of uses. Now, some of the things that we use microbes for in industry are making yoghurt, cheese, vinegar, silage and compost, as well as brewing, which we'll look at a bit later. Now, three of those processes are on this slide here. We've got yoghurt making, cheese making and vinegar. Now, the bacteria that are involved in these processes for cheese and yoghurt is lactobacillus, which is also used in making silage as well, and for making vinegar is acetobacter. So we'll look in more detail at the process of making yoghurt later on in this video. It's been made for over 5,000 years. As mentioned earlier, they use lactobacillus, which they add to milk, and it's very nutritious, and that's why it's so popular. It contains protein, calcium and vitamins, which can be eaten as part of a balanced diet. Now, another process using lactobacillus is cheese making, which has been around for 5,000 to 8,000 years, very long time. Now, the milk is curdled in this process, and that curdling process produces a solid and a liquid, the solid being called curds and the liquid whey. Now, the way it's curdled is by adding enzymes and lactobacillus to milk. Now, they'll add protease enzymes to work on the proteins within the milk. Those solid curds then can be taken and turned into cheese, and this is a popular food and has been through history due to the fact that it contains proteins, calcium and vitamins again, which are useful for a healthy balanced diet. Now, vinegar again has been used for a long time, for about 5,000 years. Now, this is carried out by acidifying wine, cider or beer to produce wine, cider or malt vinegars, e.g. wine vinegar, cider vinegar or malt vinegar. And as mentioned previously, this acidification is carried out by the bacteria called acetobacter. Next, we'll look at silage and the production of it. Now, this occurs naturally, but is sped up by lactobacillus bacteria, as we looked at with cheese and yoghurt. Now, unlike the other three processes we looked at in the previous slides, silage making is a lot more recent in its use. It's carried out by any green cooked vegetation being piled and covered in plastic, which we can see here in this picture. This is broken down by the lactobacillus bacteria undergoing fermentation or anaerobic respiration. Now, composting we looked at in Unit 4 Biology, and this is where dead animals and plants are put into a compost bin such as this, and they're decayed naturally by bacteria and fungi within the environment. Now, this forms a nutrient-rich soil which can then be put on plants and crops, especially when it's in use in organic farming. We're now going to take a closer look at the yoghurt making process, where this bacteria lactobacillus is used. So first of all, as we know, yoghurt is produced from milk. The milk is pasteurised, which means that it's heated to around 71 degrees Celsius for 15 to 20 seconds. This kills most of the bacteria. Now, this step is really important, that we keep this process sterile, free and clean from microbes to prevent any spoilage or disease being spread. Now, at this point, bacteria that are added at a stage here that we'll talk about later multiply using the milk as a food source, so we get rapid reproduction and multiplication here. Then the lactobacillus that have reproduced and replicated will break down the lactose, which is the sugar found in milk, into lactic acid. This is what gives yoghurt its sour taste, and this process is where the milk is converted to yoghurt. And when that's happened, we can add flavours, colours to take away this bitter taste of the lactic acid, and the product can then be marketed and sold. However, this is not the end of the story, because after this point here where the yoghurt is made, we can take a sample to check the quality. Now if the quality is high, that sample can be added to the next batch, and then you're keeping the quality of this sample that you've taken, so we'll get high quality yoghurt here, and you know that this bacteria are going to multiply and produce the yoghurt to be able to sell it and market it. Now we've just looked at some of the ways that bacteria are really useful to us. However, fungi are also useful in making food and drink, and yeast is a fungi that we use to make alcohol in the brewing process, and that's what we're going to look at next. Now the way that this happens is that the yeast will ferment the sugars found in fruit or grains. Now wine is used by fermentation of the sugars in grapes. Beer and lager by the malted barley, and cider is from apples. So first of all, we need to look at the stages of beer making or wine making. So here we have the process of brewing, from the malting in the process all the way down to pasteurising, packaging and bottling. Now the malting process is where the barley seeds, when it comes to beer and lager making, start to germinate. Now this germinating process converts any starch that is stored in the seeds into sugar. Okay, now it's this sugar that will be available to the yeast to ferment to produce the alcohol later. Now this sugar is needed to be extracted, so the plant material is either crushed or soaked in water, and this will get the sugar out of the grain. Now alcoholic beverages get their flavouring from, for example, wines get it from the grapes, the fruit juice from the grapes. Cider will get it from the apple juice from the apple. But beer get the flavour from hops, which is the female flowers of the hop plant, which this is the Latin name, Humulus lupulus. Now at this point, when you've got the sugar available for fermentation, we add the yeast is added and the container is sealed. This prevents any air and other microbes entering to keep it sterile. Now at this point the yeast will respire aerobically just for a short amount of time, because any oxygen that is left in that container, which there will be used up. Now this allows the yeast to use the oxygen to respire, reproduce. The liquid is kept warm so that this can happen, so we'll get the amount of yeast increasing, so we'll get more yeast to produce more alcohol. Now after a short period of time we'll start to get fermentation. This is due to the fact that this culture will quickly become anaerobic, because the yeast will use up any oxygen in respiration, and at this point, when it does become aerobic, it will start to produce alcohol. However, it's really important to note that if too much oxygen is in the container, then the respiration, the anaerobic respiration, will not occur, and it will produce vinegar. And the process happens on a large scale in factories, so really big stainless steel vats, which are called fermenters, which is easy to remember. It's kept at a constant temperature of 25 to 30 degrees. That will give the best rate of respiration to produce alcohol for the enzymes within the yeast without denaturing. The temperature also gives the alcohol a better flavour. Now when this is finished, when this process is done, the liquid is separated from the yeast cells. We do not want the yeast cells to be left within this alcohol. Now the way that this happens is by a process called flocculation, where the yeast sink to the bottom of the vat and they are able to separate that from the liquid. Now usually before it's marketed, it is clarified by filtering. And the last stage is the pasteurising and packaging stage. Now the alcohol that is produced is heated very quickly and cooled, and this will kill any microbes that could spoil the product or cause any disease if if consumed by people. By doing this, it gives the alcohol a longer shelf life when it's stored in bottles. Now you do need to know the starting products of alcoholic drinks. We've looked at cider, wine and beer before. Cider, the source being apples, wine was grapes, beer and lager was malted barley. However, we didn't look at rum, whiskey and vodka, and we're going to look at that now. Now these are slightly different because these are what we call spirits, and we'll look at how they're produced very shortly. Now rum is produced from sugarcane, whiskey from, as with beer and lager, malted barley, and vodka is actually produced from potatoes. So as you should already know, this is the word equation for the process of anaerobic respiration in yeast. We've got glucose, which is a sugar, and it is broken down into ethanol, which is the alcohol, and carbon dioxide gas. This is the symbol equation for the process of anaerobic respiration. We've got C6H12O6, which is glucose, fermented to 2C2H5OH plus 2CO2. The 2's at the start of these two molecules show that there are 2 molecules of this. So here there are 2 molecules of C2H5OH and 2 molecules of CO2. Now there's a limit to the concentration of alcohol that can be produced by the brewing process. Alcohol is actually a waste product to the yeast, and it's toxic. It will eventually kill the yeast in the fermenter when the alcohol reaches a certain concentration. This is just like us having to get rid of carbon dioxide or urea or anything like that, because if they get too high, then those concentration of those chemicals can be fatal to us. However, some yeast can tolerate more alcohol concentration than others, and this results in drinks with different alcohol contents. So beers and lagers are usually about 3 to 5%, whereas wines are usually about 11 to 12% alcohol. So the yeast that produce wine will have a higher concentration limit to alcohol than the ones that produce beer and lager. Now, spirits such as vodka and whiskey contain really high levels of alcohol, and to make these spirits, the first step is fermenting plant material, similar to what we've looked at earlier when it comes to the production of beers and wines and cider. But as we said before, rum is made from sugarcane, whiskey from malted barley and vodka from potatoes. Now spirits contain typically around 40% alcohol. Now this is a lot higher than even wines that we looked at, which we said contained around 11 to 12% alcohol. This is almost 4 times as much as what wine contains. Now to be able to increase this concentration, the liquid that is produced from fermentation has to be distilled. Now to be able to carry out that process, the liquid from the fermentation process is put into a still, which is a large container. It's heated and water boils at around 100 degrees Celsius. However, alcohol boils at 80 degrees Celsius. This is lower than water. So, the alcohol will boil first, rise up the column as vapour, leaving the water behind. That vaporised alcohol will pass along the collecting arm, cool and be collected, and we've got a lot more alcohol, a pure alcohol from that fermentation liquid.